Hey, it's Natalie. It's been a little while. I screwed up the Mimi, Alex, Natalie pattern, but I'm back. And for this episode, I spoke with Christine Sprunger, who is head of the Soil Health and Ecosystem Ecology Lab at Michigan State University. She's a soil scientist who has done or is doing a lot of the things that are really important to understand what's happening in the soil, these below ground processes like what's going on in the rhizosphere. But these things are things that others have shied away from because they are so painstaking, like studying roots, or now she's studying nematodes. She's been working closely with farmers on soil health and soil health assessments, and she's actually currently seeking farmer collaborators for upcoming projects. So check out her lab's website in the show notes and reach out to her lab manager if you're interested. She even said you get a free soil health assessment.
Today's episode is brought to you by Real Organic Project. Does your farm deserve to be recognized for all the hard work done on behalf of the environment and your community? Real Organic Project is an add-on certification that partners with 1,000 plus certified organic farms across North America, including my own farm, Rough Draft Farmstead. Available at no cost to farmers and with minimal time commitment, it is a great way to differentiate your farm from mass-marketed corporate organic, where hydroponic production and animal confinement are still commonplace. Real Organic Project is a whole farm certification program to distinguish crops grown in healthy soils and livestock raised humanely on pasture. As a farmer-led movement, we know many hands make for quick work. I hope you will lend yours by signing up for Real Organic Project certification today. Visit realorganicproject.org/no-till to apply. That's realorganicproject.org/no-till to apply. Today's episode is also brought to you by Johnny's Selected Seeds. Johnny's Selected Seeds is a proud sponsor of the No-Till Market Garden podcast. Since 1973, Johnny's has supported farmers and gardeners with superior seeds, tools, and information to help ensure their growing success and help feed their communities and families. The research farm is at the heart of Johnny's, where they trial thousands of varieties and tools every year. Their breeding program uniquely focuses on introducing varieties that meet the needs or solve the challenges mixed market, small farmers, and avid gardeners face. Visit johnnyseeds.com for innovative new varieties and time-tested favorites to grow this season. You can also browse their online growers library, link in the description, for a wealth of free educational resources. The employee owners at Johnny's look forward to growing with you. All right, enjoy the show.
It's great to have you here. I'm really excited to talk about nematodes, among other things, today. And so if you could just introduce yourself, where you are now, what is your job description, and what drew you to this field of research?
Yeah, so my name is Christine Sprunger. I'm an assistant professor of soil health at Michigan State University, and I'm based at the Kellogg Biological Station, which is about an hour and 15 minutes south of East Lansing's main campus. It's a small research station, but what's really great about being based at the Kellogg Biological Station is it's home to about 3 different long-term agricultural trials. So we can ask a lot of really important questions within the space of regenerative agriculture and soil health.
And what drew you to this field in the first place?
Yeah, so I think growing up, I was a child that always liked to be outdoors. I grew up in Washington State, and so I was really drawn to forests and the ocean. But when I was an undergrad at the University of Washington, I randomly took a soils course. And I was like, wow, this is exactly what I've been looking for, because it is so critical for food security, but also is critical in alleviating a lot of the major environmental issues that we have. And so I loved kind of the duality of soil science. And so I knew that I wanted to go to grad school. So I went into a graduate program right out of undergrad, and I actually did my PhD at Michigan State University. So it's nice to come full circle and now be faculty here at Michigan State.
And when you were an undergrad and you took that soils class, did you even know that soil science was a thing?
No, I didn't know it was a field of study. I knew that soils were important, and I was drawn to kind of different like soil conservation efforts in Haiti. That's actually where I was born, and so I was always kind of conscious of soil erosion type issues, but I had no idea that people could literally devote their entire lives not only to soil science, but that there are different aspects of soil science, so soil biology and chemistry, etc. And so that just kind of like really just got me excited about learning everything I could about soils.
Me too. I had a similar experience as an undergrad. I took Intro to Soil Science, and I remember just seeing the soil pits and like getting that three-dimensionality to soil for the first time. Really, I was like, whoa, there's more to soil than what we see. And there was no going back. I also fell in love with soil after I took my first soil science class. So that's awesome. I am curious. I know I didn't put this in the outline, but one of the things that when we were talking about doing this interview that you mentioned was you have a research appointment and you don't have an extension appointment. And I was wondering if you'd be willing. I think a lot of us in the land grant university world, we speak this language of appointments, and people are like, "What the heck are you talking about?" And they just expect your professor at a land grant university. This is what you do. So if you could, so first of all, thank you very much for being on the podcast, because I know you don't have an extension appointment. But if you could just talk a little bit about that difference between a research appointment and extension appointment, how people split that at the university, and how just for farmers to understand too, when you're reaching out to people at the university, why do some people seem to do a lot of outreach and other people don't do any outreach?
Yeah, no, it's a really good question, and it's a kind of confusing answer. So bear with me. So, especially within any kind of college of agriculture that's based at a land grant university, there is a wing of extension. So farmers are likely having the most interaction with extension educators. But there's also a subset of faculty, so professors at your local land grant university that have an extension-based appointment. And so by, part of their funding dollars are accruing through the extension kind of platform, and they are mandated essentially to devote 30-50%, whatever it's allocated, to efforts surrounding extension. So that could be doing on-farm research, or it could be going to specific farmer centric conferences and giving talks. And then another portion, another kind of section that you might be mandated to work on is research. So most professors have some type of research appointment at land grant universities. In my case, it's 70% of my role. And so that just means that I am evaluated on, publishing papers and bringing in grant dollars. And then the last kind of wing is the teaching component. And so for me, it's actually 20%. And you'll notice that there's 10% missing from that. And so that 10% that's missing is service. And service can come in a wide range of kind of required service to the department. So I serve on committees, and I do outreach and that kind of thing. So that kind of all counts within service. So, for example, your state soil fertility specialist likely has a large extension appointment, and they are often the person that you go to with any type of soil based question that you might have. But it's also important to know that there are other faculty who, even if we don't have extension appointments, we're still very open to talking to farmers. And we even do on-farm research. I conduct on-farm research quite often, even though I don't have an extension appointment.
Yeah, thank you for that explanation. That was really helpful. So now let's dive into the science. And I wanted to start out with what I know the least about, and that's nematodes. So I was wondering if you could just tell me all about nematodes and also the story of kind of how you became interested in nematodes also. Yeah, just go for it.
Yeah, so nematodes are super exciting because they are microscopic, non-segmented roundworms that are very plentiful in the soil, and they're also found in freshwater ecosystems. And what's really special about nematodes is that they're specialists, so that there are some nematodes that only feed on fungi. There are some nematodes that only feed on bacteria. And then there are some nematodes that feed on plant roots. So those are your plant parasitic nematodes. And then there are also nematodes that actually feed on each other, and they're predators and carnivorous. And so based on kind of the different ratios of these different types of nematodes, we can actually tell a lot about soil health and various nutrient cycling. For example, bacteriivores, so nematodes that only feed on bacteria, are often found in systems that are really heavily disturbed. So if you have an agricultural system, for example, that you see just a lot of bacteriivores, you understand that likely that soil health there is, not as healthy as a system where maybe there are more fungivores present or more predators. And so what we're trying to do is understand kind of the linkages between nematode communities and various soil health kind of outcomes. And the reason why I even went down this kind of nematology route, because I wasn't even trained in nematology during my graduate work. I started this as a faculty member when I was at Ohio State University. It was all because farmers were asking me for soil health indicators that directly measured soil organisms. So I took time to really think about, which organisms made sense to study and really integrate within the soil health framework. And for me, nematodes are kind of a no-brainer. And the reason for that is because of their position in the soil food web and because of kind of the downstream effects that they have on bacteria and fungi and nutrient cycling, we can link them to kind of important ecosystem processes. It's much harder to link bacteria and fungi, for example, to nitrogen cycling and carbon cycling, by specifically seeing, this certain type of bacteria. It's often hard to make that link. But with nematodes, it's a little bit easier just because they are slightly higher up the soil food web.
So one of the things that pops into my mind is if you could also talk about how you extract them from the soil and then look at them and identify them. And then relatedly, I know that one of the sort of problems that microbiologists have is when they extract DNA to sequence who's there, you don't know whether those microbes are active or not active. So when you're pulling out the nematodes, are you only getting living, active nematodes?
Yes. Such an important distinction. So for nematodes, there are multiple different ways that you can extract them from soils, but the way that we extract nematodes is using a Biermann funnel. So all that is we place 40 grams of soil in filter paper. You could even use like a Kleenex if you wanted to, and we place them in funnels. And then the funnel, at the end of the funnel, we have just a tube, a plastic tube. And then we kind of clinch that tube and then we basically water that funnel. And so what that does is the nematodes are kind of drawn to the bottom of the funnel and into the tube. And as we water them, they gravity does its job and the nematodes flow to the bottom of the funnel. Then after 3 days, we release the clip on the tubing, and then we use a beaker, and then the nematodes just flow through with the water that's in that tube. And so from there, we then just put that solution of water into a petri dish, and then we are able to count and identify with a microscope the different types of nematodes, and they are living. We usually preserve them because it's easier to identify them
if they're not moving.
Exactly. And the way that we identify them is they have very important mouth parts that tell if they prefer to eat bacteria or fungi, for example, but then they also have other kind of body metrics, things like longer tails or one or two gonads. Those are ways that we identify them into families and genus. And so based on those placements, again, we can tell if a system is really disturbed or a system was high in nitrogen, because certain nematodes only adapt to certain types of environments.
And how long, I'm just curious, how long does it typically take to go through that process of looking through the microscope and identifying all of the nematodes from one 40-gram sample?
Yeah. And so the labor and time is the limiting step. And this is why a lot of people don't do nematology. My grad student tells me it takes about 2 to 3 hours to do one sample, which is a lot of work. And, depending on where the sample comes from, if it was like coming from a polyculture system or a prairie system that has a lot of diversity, there can be like thousands of nematodes in a given sample. And so there are, of course, like metrics and ways in which, for example, we only count 100 of those 1,000 because we know it's a representative kind of account and identification, but it still takes a lot of work.
And are you looking at sort of abundance and diversity, or just diversity? Or
Yeah. So we like to do abundance just to understand kind of sheer number of nematodes in a given sample, but doing abundance is not just abundance solely is not enough. What really gets us to be able to understand more about soil health in that system is by identifying and understanding the different ratios of the nematodes. And that's just a crucial step in understanding kind of the nematode community composition and how that is reflecting, overall ecosystem health.
I had one other question about nematodes before we talk about your specific research with nematodes. I know that a lot of people, well, myself, I'm assuming other people too, have heard of nematodes most in the context of the plant parasitic nematodes, but then also in the context of the beneficial nematodes that you can buy in a bottle. So can you talk about what those beneficial nematodes are and how they fit into the system and how long they might stick around if you buy a bunch of them and put them in your soil?
Yeah, so certainly the most common nematode that farmers and really the general public are going to be familiar with are soybean cyst nematodes, and those are obviously detrimental for crop production and very parasitic. The nematodes that we're mostly interested in are beneficial nematodes, and these are naturally occurring nematodes that are found in our soil. And depending on the ratio, like I mentioned before, they can tell us a lot about how healthy your soil is. A separate component is, sure, you can go out and buy beneficial nematodes and apply those onto your field. But in all honesty, from just what I've read and kind of where the research is at, I'm not sure what the scientific benefit is in terms of, do you see greater yields? Do you see overall greater health when you're adding, these kind of custom made nematode mixtures? I just think that more research is needed in that space.
All right. So moving into your research, you've done a lot of work. I didn't know that you actually started doing the nematodes later. I thought you had started doing that in your graduate work, and that's really neat that was kind of a farmer-inspired thing. And I think it speaks really to the fact that, even as a research faculty member, you are still very much listening to what farmers' interests are, and that's really cool. So you have done some work building off of long-term trials in Michigan and in Ohio. Can you tell me about your past and ongoing research projects in some of those systems and what you found?
Yeah. So, as people are likely aware, some of the big questions that we're asking around regenerative agriculture, sustainable agriculture, whatever you want to call it, are things about how carbon changes, which systems are most effective at accumulating carbon, how nitrogen might shift, whether or not you have no-till management, or whether or not you're including a cover crop. The responses to these management practices often take a lot of time. And so that's why these long-term trials are often established. So the no-till plots that we were working with at Ohio State University, those were established, my goodness, in the 1960s, I think. Yeah, so that's super long time ago. And then the trials that we have here at the Kellogg Biological Station, the longest one was established in 1989. And so what we can really learn from these long-term trials is how long it takes, for example, for cover crop implementation or no-till implementation to really give you those multiple benefits that you want to see on your farm. And so, again, the questions that we generally are asking are soil health-based questions. And so, for example, in the KBS LTER, we're really wanting to understand which treatments are influencing carbon cycling and whether that carbon is really staying in the ground, if that's like a permanent kind of carbon sequestration, which is important for mitigating climate change, but then also it's really important for long-term soil fertility and improving soil health. So again, it has kind of those dual benefits. And what we're finding is that systems that have some type of diversity and a component of perenniality too are really important for moving the needle in terms of overall soil health.
And specifically with nematodes, have you looked at how those treatments, and if you get into those treatments a little bit more, how those have affect the nematode communities?
Yeah, so actually my grad student, one of her dissertation chapters is comparing nematode communities that were, counted and identified back in 1992 from the KBS LTER, and then comparing it to 2021 when she did the sampling herself, and she extracted the nematodes the exact same way. And she's finding some really interesting results across the treatment. So there are like 8 treatments within the KBS LTER, but it's most, we can generalize the treatments in kind of annual row crops, monoculture perennials, and then diverse perennial systems like prairies, for example. And what she's seeing is that over time, in the 2021 sampling, there's a greater ratio of fungivores relative to bacteriivores, especially in those perennial and more diverse systems relative to the annual system. Whereas in 1992, all of the treatments were basically the same, and they were dominated by kind of bacteriivores. So that's telling us that long-term implementation of kind of regenerative practices are actually shifting the nematode community in what we see as a more kind of beneficial nematode community that could be contributing to greater overall ecosystem services like greater overall carbon accumulation and greater nitrogen retention and all those good things that you want to have happen in your field.
That's really interesting. And that kind of leads into my next question. which is, so nematodes have historically been studied kind of in isolation. And you're working on, I understand that you're trying to make those connections between the nematode community and the microbiome or the microbial community. And I'm wondering how you're approaching this and what you're finding and why making these connections are important.
Yes, absolutely. And I'll add another thing that we're trying to integrate. We're trying to integrate all of the other aspects of soil health too. So aggregate stability, enzymes, all these various indicators that people might have heard of. So from our findings right now, what we can say is that the nematode feeding groups, so things like bacteriivores, fungivores, predators, etc., they actually integrate really well with soil health indicators like soil respiration. So a lot of farmers might have heard of like the Solvita test, the burst of CO2. That is actually highly linked with some of these beneficial nematodes. ACE protein, which is a soil health indicator that reflects nitrogen, also looks to be really highly linked to some of these nematode communities. Interestingly enough, we also see kind of the plant parasitic nematodes are also kind of associating with that burst of CO2, but that's showing that it's kind of a mineralization process. So, these nematodes are feeding on the roots, and those roots are turning over and leading to more of a burst of CO2. Now, whether or not that carbon is actually staying in the soil or actually leaping into the atmosphere has to be done via other tests. But it's just interesting that those linkages are there.
That kind of brings into question, okay, what is the value of the CO2 burst if it's has these correlation with both these beneficial nematode community and this parasitic nematode community? I interpret that as, okay, you can't replace our studying the nematode community with the CO2 burst. So can you just talk about that a little bit?
Yeah, no, that's a really good point. So with all these indicators, we study them in isolation, but we also need to understand how they're related to one another to really understand kind of the actual soil health outcome. So for example, we see that fungivores, so nematodes that are feeding on fungi, are really closely related to permanganate oxidizable carbon, which is a soil health indicator that reflects a more processed pool of carbon, and it could even reflect a more stabilized pool of carbon. And so what that tells us is that when we see more fungivores, perhaps we are seeing a system that is accumulating carbon in a way that could lead to long-term soil carbon storage. In the reverse, if we see beneficial nematodes with that burst of CO2 and that mineralization process, that could mean a couple of different things. It could mean that, sure, maybe that carbon is leaving the system, which we don't want to see, but maybe it actually is a reflection of nutrient release, which could be really good for the crop. And nutrient uptake and crop productivity. And so, again, these are indicators, and then we need to understand how they're integrated to really understand the entire system and the overall benefit to the field and ultimately to the farmer.
Do you think, this is jumping ahead a little bit from where I had wanted to be, but do you think that we will be able to use these, well, so both permanganate oxidizable carbon, also known as active carbon, and respiration are both really chemical measurements trying to get at biological processes. But do you think that to get at the biology, we actually need to have some biological measurements?
That's what I'm a proponent for. I mean, one of the kind of foundational cornerstones of soil health is increasing soil biodiversity, and none of these indicators can actually reflect that except for nematodes. And so the benefit that I see from nematodes is that they can reflect these key kind of chemical processes that we're seeing, but then they can also demonstrate the overall diversity of the soil food web. And so obviously we need both to have a kind of rigorous soil health assessment. But I do think that nematodes are essential if we're really going to use soil health for biodiversity metrics, especially.
I think I interrupted you when you were starting to talk about how that relationship between the nematodes and the microbiome and how you're going about studying that. So we can get back to that. I apologize for the interruption.
No, yeah. And so that's the aspect of linking nematodes to various popular soil health indicators is one aspect of the work that we do. But we also actually, in terms of the soil biodiversity metrics, we want to understand how nematodes are actually influencing bacteria and fungi. And so you'll notice that I've said, that nematode is a bacteriivore. It feeds on bacteria. That we don't actually know which bacteria nematodes prefer. Is it a bacteria that's really important for nitrogen cycling? We don't know. We have zero idea. And so we're working on making those linkages. And it's tough because we need a very controlled environment to be able to, really assess which bacteria, for example, are most influenced by a specific type of nematode. So I have a grad student that's kind of working on different controlled environments where she's going to build soil columns, and then she's going to add different rates of fertilizer, and then she's basically going to watch kind of what leaches through the soil column and then collect the bacteria and nematodes that are in that sample collection and really see, what that community looks like and how they're related to one another.
That's interesting. So you're saying there's a really basic science gap.
Yeah.
That's interesting.
Exactly.
Okay.
Yeah. Yeah. And the gap between fungivorous nematodes and fungi is even more confusing, and we know even less. And so again, those are really kind of basic scientific questions that we're working towards. So that's another reason why I really like nematodes, is because there's some really key applied questions that are really important for farmers. And then there are kind of these kind of basic science questions that are just really important for advancing our understanding of nematology.
Yeah. You said that you identify them by their mouth parts, among other things. I know if people go online and you Google, different nematodes, there are very cool images that come up. But could you talk a little bit about the mouth parts and just how they differ? Because it's kind of a neat evolutionary story, I guess.
Yeah. So for the predators, the ones that are actually feeding on other nematodes, there's just like this massive tooth that's pretty visible through the microscope. And then for the plant parasitic nematodes, the plant root feeders, they have a spear that they just kind of jab into the plant root. For the bacteriivores and fungivores, the mouth parts are kind of hard to describe, but they're pretty distinct. There's kind of just like a hole for one and like an oval shape for another. But those mouth parts are critical in how they're able to consume their food. And then actually there's another set of nematodes that I haven't mentioned, and they're the omnivorous nematodes. And so based on the environment that they're in, they'll actually shift and feed on whatever they can.
So do they have multiple mouths?
They generally have kind of an oval shaped mouth. So they're generally not predators, but they will kind of shift between bacteria and fungi.
Today's episode is brought to you by Rimmel Greenhouses.
Whether it's tomatoes to market, flowers in the spring, or your family's harvest, growing in a greenhouse protected from the weather provides the right environment that you need for a harvest that you can count on. Rimmel greenhouses are designed for durability and offer the quality that you need to grow productively year-round. Visit rimmelgreenhouses.com to get a quote today.
If I may editorialize for a second, I own a Rimmel tunnel. I love it. We built it ourselves, and I really appreciated the quality of the tunnel as well as the customer service we received in the process. So make sure to check them out. Today's episode is also brought to you by Tilth Soil. This Cleveland, Ohio-based company is producing some of the highest quality potting mixes out there. One of the biggest lessons I learned in farming is the importance of good seed starting mixes, and that's why I included Tilth Soil in my book and have been using their seed starting mix, Sprout, on my farm since 2020. Tilth Soil produces potting mixes approved for use in organic operations like our own. Their living soil is made with fully composted food waste they collect and process themselves. Whether you need a cubic yard super sack once a year or ongoing deliveries for your year-round operation, Tilth has you covered. The team at Tilth can help with shipping, coordination, and provide ongoing support throughout the growing season. To learn more, visit www.tilthsoil.com. That's tilthsoil.com. All right, back to the show.
I know that there are folks who feel like soil fauna really gets overlooked. What are the advantages of studying nematodes versus, and I'm not saying we should do this in exclusion, we should do it all, but versus studying insects and that whole community?
Yeah. So, yeah, I feel like it ultimately depends on the soil health outcome that you're interested in, or the environmental benefit that you're interested in. If you're interested more in, pollination and kind of just thinking about above ground processes, insects are really important, and thinking about just higher up the overall soil food web, I guess, or just the food web in general. But with nematodes, I think they're so central, and they could actually be a link to kind of up the food chain, because mites feed on fungi or on nematodes, and so that's just a clear link to above ground insects. But I think really it's the placement of the nematodes in the soil food web. They're just so trophically diverse. And the fact that we can kind of differentiate by feeding group really, again, is what is helpful in linking them to overall kind of ecological function, whether that be nitrogen cycling, carbon cycling. And so that's kind of why we see nematodes as important to focus on.
Yeah. Moving in a different direction. So nematodes are pretty laborious, but exciting to study. And you have also done work with roots, which are really laborious, but also exciting to study. So I was wondering if you could talk a little bit about your work with below ground biomass, roots, and how you've been trying to use that to understand the drivers of below ground carbon and nitrogen cycling. And you can start with why it's important to study roots.
Yeah. I feel like, so roots were really the premise of my dissertation work. And oftentimes when we think about carbon cycling and nitrogen cycling, we think about the things that we can see, thinking about overall crop productivity. And of course, soil is a major factor too, but we think less about the biomass that's below ground. And roots are really important because they actually are contributing more to soil carbon than above ground. For example, we know that carbon derived from roots persists in the soil about 2.5 times more than, say, an above ground piece of residue that's decomposing. And so understanding what's happening below ground is critical for understanding overall carbon sequestration potential for a given system.
And what about nitrogen?
Yeah, nitrogen is also key too, because roots are basically the stopgap between the nitrogen that is staying in the soil versus flowing into our groundwater. So some of the work that we've done has been comparing annual winter wheat to perennial intermediate wheatgrass. It's a perennial crop that's being bred at the Land Institute. And what we found is that roots of these perennial intermediate wheatgrass systems are essentially reducing all nitrate leaching from the system, which is fantastic relative to annual winter wheat, where we see a lot of nitrate leaching. And that's not even as much as what we'd see in a corn system, for example. And so these deeper, extensive roots that you see in perennial-based systems are critical for nitrogen retention and carbon accumulation.
And can you talk a little bit about sort of the different allocation strategies of perennials versus annuals and why that might be?
Yeah, so that's a really good point. And the reason why you're able to see these kind of ecosystem services within these perennial systems is that because plants are perennial, they're wanting to survive for the long term. And that survival is really stemming from their root systems. And so they're allocating more biomass below ground. Oftentimes we see, 3 to 4 times more root biomass. Sometimes it's even up to 10 times more root biomass, depending on the perennial crop relative to annual counterparts. Annuals, on the other hand, they're around for one growing season. And so they're going to allocate their energy above ground. And so they have much less roots. And so again, it's often a trade-off, right? You have greater yields typically from annual systems, but a lot more ecosystem disservices, whether it be losses of nitrogen or just lower overall soil health. Whereas in perennial systems, sometimes you can still get a yield or you can graze, but you also have those really key ecosystem services.
Yeah, and you mentioned when you were talking about the long-term experiments, that they kind of have this gradient of perenniality. And I think people use this term perenniality in two ways and can be a little bit confusing. Perenniality meaning there are roots growing in the ground all year long versus perenniality, you're growing perennial plants. Can you talk about just the benefits of the first kind of perenniality and just kind of that distinction between those two and what you might see from quote unquote perenniality in an annual system versus perennial system and integrating annuals and perennials and wherever you want to go with that.
Yeah. So I'll answer it by describing another long-term trial that we have here at the Kellogg Biological Station. It's part of the Department of Energy funded biofuel cropping system experiment. But there we have this beautiful kind of diversity gradient where you have annuals, like continuous corn, then you have corn in rotation, and then back when I was doing my dissertation work, we actually had systems with corn plus various different cover crops. So that's your first kind of version of perenniality, where you're actually having, dominated annual systems, but because there's a cover crop and because of an extensive crop rotation, you have ground cover for most of the year. So that is a version of perenniality, especially within agricultural systems. Then you move into your monoculture perennial systems. So that could be intermediate wheatgrass, which I mentioned before, but in this specific trial, it was candidate biofuel crop switchgrass and miscanthus, and then you move into more of your diverse polyculture systems, so things like native grass mixes and restored prairie systems. And what we see is that even just within the annual row crop systems, just by adding a cover crop, you do see a bump in soil health. You see greater overall soil carbon storage and those other kind of indicators that we talked about earlier. But you actually start to see even a bigger bump in soil health when you move to your monoculture perennials. And then you actually see 2.5 times more of the, things like soil respiration and pox relative to annual systems and near more diverse polyculture systems. So it really is this gradient, and it's this gradient of not only annuals versus perennials, but also just increase in overall diversity that's really important.
And I think I read one of your papers with one of your grad students talking about the importance of perennial legumes too, specifically. Can you talk about the role that perennial legumes play and why they're so critical towards boosting up that soil health, and in particular the nitrogen cycling piece?
Yeah, absolutely. So not every farmer is going to want to experiment with switchgrass or miscanthus, but there are a lot of farmers that might want to have alfalfa in their rotation. And so one of the long-term trials that we participated in at Ohio State University looked at just that, just looking at corn soy rotations, but also having an alfalfa in the alfalfa crop in the rotation, and having that perennial legume was critical for maintaining the nitrogen pool and really boosting soil health overall. And again, that's coming from having deeper roots. We actually measured fine roots within the corn system, but we found a higher overall nitrogen within the fine roots, and we think that's a legacy effect from having had the alfalfa within the crop rotation. And so it's really, just imperative to have that, extension of the crop rotation with that perennial legume for enhanced overall soil health.
And that's something that kind of fits if you have a diversified rotation. It doesn't necessarily have to be alfalfa. It could be clover. There are a number of perennial
legumes. It could be hairy vetch. It could be, yeah.
So let me see where we are here. We've talked a lot about soil health, and a lot of people are talking about soil health, but there's still a lot of ambiguity about what that means, how it's measured, and then importantly, how the measurements influence management decisions. So it came back with this number. Okay, well, what does that mean? What should I do? So could you tell me about the work that you're doing to bridge those gaps between farmer perceptions of soil health, the things we can measure about soil health, or what we call soil health indicators, and then where you think it's going in the future? And I know you've already said that you're an advocate for including nematodes as a soil health indicator, but could you talk about just bridging that gap and what is soil health?
Yeah, so sure, there are a lot of kind of academic arguments around what soil health actually means, and I kind of think of the standard definition from the National Resources Conservation Service, NRCS, and I have it right here. Soil health is the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. And I think that's like the clearest definition of soil health, but ultimately I don't think it matters how we describe it. It matters how we actually quantitatively measure it. And so that's thinking about the indicators that are most useful. And ultimately, I think it comes down to the farmer. It really comes down to what outcomes the farmer is most interested in. Are they interested in building soil carbon? Are they curious about their pools of nitrogen? Do they want to understand kind of the biodiversity that's on their farm? That's ultimately what's going to, I think, dictate which soil health indicators should be run on a given farmer field. And that's kind of the approach that we've taken in terms of really wanting farmer feedback on soil health indicators, because we can measure, 20 plus indicators all day, every day. And really, is that going to push the needle on improving soil health on farmer fields? I don't think so. And so one strategy that we've done is we've actually worked alongside farmers in Michigan and Ohio. And we've actually asked farmers to pick different fields that they're curious about on their farm. One popular method that we use is we just ask them to pick a field that they think is their best and then a field that they think is their worst. And then we measure, a suite of soil health indicators, whether they be biologically based, chemically based, or physically based. And then we kind of draw up a soil health test report for farmers, and we walk through it with farmers. And we ask them if that makes sense. Do these indicators kind of match up with your perceptions of your field? And sometimes they do, and sometimes they don't, which is problematic. And that's where we learn more about, are these indicators sensitive enough to pick up some of the issues that farmers are facing on their fields? And what I've really come to realize is that farmers are most interested in indicators that are more sensitive and that can actually pick up the differences between their best and worst fields. And some of those are things like soil respiration. I think soil respiration does the best job at picking up differences between their best and worst fields kind of across the board. But one of the things with soil respiration, it is also really variable. And so it's kind of hard to trust that number. We have to remember this is just a snapshot in time, and that it could shift within a week. And then I think farmers are also really drawn to, again, indicators that are directly measuring soil organisms. And so farmers have expressed a lot of interest in nematodes, and also things like enzymes that are reflecting biological activity. And what we're seeing, again, is that these biological indicators generally trend on reflecting what the farmer is feeling about a given field.
And so what I'm hearing you say, and I think this is kind of how I think about it too, is that soil health is valuable from almost an individual farm perspective of like changes with time and changes with management, but comparing from one farm to another is not necessarily that helpful. And so the analogy between sort of human health too, like you could go and say, how do I get healthier? But it's largely a matter of, well, what do you care about? And let's measure, I don't know, if you want to be an endurance athlete, then that's going to be a different set of things than if you want to be a bodybuilder or whatever. The analogy doesn't totally hold, but just that soil health is more about, let's see how things are doing, and then and pick a suite of things that you care about, and then monitor that with time, or see how it changes when you do this or that, and come up with a unique management plan for each farm.
100%. And, sticking with kind of the human health analogy, just like health professionals are running clinical trials, etc., soil scientists need to do that as well. And so we're kind of building a soil health database to where we can be able to understand what a given soil health test on a farmer field is relative to, farms across that given county. And so, for example, right now, you wouldn't want to, compare a male wouldn't want to compare his BMI to a female, for example. And a farmer with a sandy soil is not going to want to compare their soil health to a farmer that has a more silt loam soil.
Sure.
But as soil scientists, what we're doing is we're conducting more on-farm trials. We're conducting regional-based analyses, and we're building up these databases to where we can demonstrate to farmers, we have this pool of 1,000 different soil types across Ohio. And, relative to farms with a similar soil type, you're doing really well. You're doing above average. Or, your worst field, you're right, is actually performing, in the lower 25th percentile, where it's just trending lower compared to other fields with a similar soil type. So that's what we're working towards. And I ultimately think that's where the field of soil health needs to go.
Mm-hmm. But we don't have the database yet.
I would say we don't have it yet, but we're working on it.
Yeah.
So we have, my colleagues and I in Ohio, for example, we've collected over 2,000 samples. And we have every county except 7 represented in our soil health analysis. And I want to do the same for Michigan. So we're headed there, but we're not there yet.
And are you going to have someone sitting in front of a microscope and do nematodes on every one of those samples?
Likely not on every one of those samples, but we'll try. We really do also want to build a database for nematodes because it's the same thing. It's really hard to compare, across all these different fields unless we know the soil type and we know a little bit about the management history as well.
Yeah, that would be an amazing database. So you have some current work that's looking at climate change, and one of the things about the NRCS soil health definition, it kind of implies it, but it doesn't really get at this idea of resilience. Like it gets at the idea of, continued capacity, but in the face of climate change, that means the continued capacity, which is different from what it was. So I'm curious about some of your current work on how climate change and in particular the drying and re-wetting events might affect soil processes.
Yeah, so we have 2 really big experiments that are focused around climate change impacts on soil health and just below ground processes in general. So our first is a big flooding project where we're working with Dr. Alex Lindsay at The Ohio State University, and he's an agronomist and plant physiologist. And so what we're asking is, how does flooding intensity and longevity of floods impact not only corn production, above ground biomass, etc., but also thinking about below ground dynamics. So we're looking at vine root production, we're looking at nematodes, all the different soil health indicators that I've talked about so far this hour. And we're really wanting to understand, again, getting at that resilience of how do these major flooding events impact the overall health of our system? And not only do we want to understand kind of that acute impact, we're measuring over the course of the entire growing season. So what we've done is that we are looking at early summer flooding events primarily. So after the corn is planted, we'll flood using drip irrigation, a wide range of treatments. And then we'll sample like after 5 days or after 3 weeks, after, right before harvest. And we're really trying to kind of put together a puzzle of what types of management practices are most beneficial in terms of kind of alleviating flooding effects, and how might farmers be able to kind of adapt to these more frequent flooding events that we're seeing across the upper Midwest. And I would say our data analysis is very early on for that project, so I can't really share what we're finding yet. We definitely see flooding effects have major influences on above ground production, and we're starting to see some indication that it's also having a big impact on soil health. The other project that we're working on is thinking about drought. So that's the other major effect of climate change. And what we're doing is we're building these rainout shelters that exclude any ambient rain that might fall during the course of the growing season. And there we're really just asking how drying influences soil health indicators. For this specific project, we're teaming up with 9 different labs at Michigan State University. So there's some people that are looking at the plant physiology aspects. There's some people that are looking at just overall crop yield. We're focused on nematodes, of course, and there are other people looking at like greenhouse gases, and we have it all covered. But what we're seeing there is that, again, the nematode community composition is really shifting when you have intense periods of drought. And my grad student is diligently completing her identification, but it's going to be really interesting to see if the nematode community is able to recover when we use irrigation to kind of alleviate the drought at the end of the growing season. And so that's kind of the recovery piece of, once you do get a rain event, does the nematode community recover from the initial drought?
Do nematodes have the capacity for dormancy, or do they have to have water? Do they need a water-filled pore to survive?
They generally need a water-filled pore to survive. What we actually are seeing is that in more disturbed systems, we actually see juveniles. So it's the adults that are unable to survive, and then the juveniles that haven't fully developed their mouth parts and other body parts are the ones that are persisting, which is a really interesting kind of overall kind of effect on the entire community.
Yeah, that's very interesting. So they're able to reproduce, but they just don't live as long in disturbed systems?
Once they produce, they just die.
Oh, so, right. So then, the next generation, what
is just continuing to proliferate.
Okay.
Yeah.
Huh. And I know you said you're kind of in the midst of or beginning of your data analysis, but what do you think are some of the things that, practical things that farmers can do to increase resilience?
Yeah, so one of our trials is specifically looking at cover crops, and we're comparing 7 different cover crops. Please don't ask me to name all of them. I have to go back and look at this spreadsheet. But we have cover crops and monoculture and then some mixtures. And we're really curious to see which cover crops are most effective at kind of accelerating the recovery. So basically what we do is the cover crop is planted, and then the cover crops are killed, and then we move into our corn phase, but then we induce the flooding event. And so basically we want to know, what is that kind of legacy effect of the cover crop? All these different cover crops have different root systems, and do the cover crops like rye that might be allocating more resources below ground, are they going to be more influential in terms of drainage and kind of alleviating flooding effects? So I'm super excited to see the results of that specific trial, but we have to be patient. Yeah.
Yes, we'll wait with bated breath. Well, I want to be respectful of your time. I really appreciate you being here with me today. Are there any final thoughts you have?
Yeah, I just want to let listeners know that if you're curious about our work, please visit our website, www.sprungerlab.com. And we, in the next kind of 6 months or so, are going to ramp up some of our on-farm efforts. And again, we want to build up that soil health database. So if you're curious in getting soil health testing on your farm, know that there may be opportunities very soon.
Oh, that's really exciting. And that's beyond Michigan or just in Michigan?
We're interested in kind of regional aspects. So, kind of, I would say upper Midwest or Northeast, we're definitely interested and open.
That's great. Well, thank you again. It was really fun to talk with you. I learned a lot, and good luck in the rest of your work.
Thank you so much.
I hope you all enjoyed this conversation as much as I did. I feel like with a lot of the guests I've had for the podcast, I want to check back in a couple years and see what new things they've learned. And of course, I want to work with all of them. But there's only so much proposal writing time in my life, and research time, and just time in general, I guess. I don't know. They're all so cool. We'll just see where life takes us.
Hey, you all, Farmer Jesse here, just jumping in real quick to catch you up on some of what we have going on. First, Hannah and I are hosting a couple small farm events here at Rough Draft Farmstead, like coming right up, one on April 25th, 2023, and the other on May 9th, 2023. There will be hands-on demos and a farm tour, and we'll dive into some of our growing systems. We will put a link in the show notes. The details are at roughdraftfarmstead.com or at the no-tillgrowers.com forum as well. I will also be presenting with Dan Kittredge in North Dakota at Minokin Farm for the Burleigh County Soil Conservation District. No idea if I pronounced any of that right, but my presentation will be on regenerative practices on a small scale. That is on May 2nd, 2023. So all the details for that will be linked in the show notes as well. Also, we asked you all a few weeks back to do a survey, and you absolutely killed it. But we have one more, much smaller survey that we would love for you to complete about no-till growers specifically. Link to that in the show notes. That is very helpful for our work. Oh, and if you haven't already, check out the No-Till Growers YouTube channel. That thing is back up and jamming again for another season with lots of very good guides and informative videos for everything from onion, tomato, pepper, and spinach production, to demystifying the tomato grafting process and simplifying compost production, all sorts of good stuff. Go over to YouTube and search for No-Till Growers and subscribe. If you loved this podcast episode and would like to support our work, you can pick up a copy of the Living Soil Handbook at no-tillgrowers.com or one of the no-till hats, which are back in stock. We've got merch there as well. The proceeds from those sales at no-tillgrowers.com go to supporting our work, which is free and open to the public. If it has benefited you and you have the means to pitch in, that would be super rad. Or even better, become a yearly or monthly patron at patreon.com/no-tillgrowers, where not only may you get discounts on our stuff, but at a certain level, or if you just bump up from one level to another, you get a shout out on the show. So big shout outs this week to Closter Farm and Livestock Co., Bill Altman, and Stephen Smith. Huge shout out to everyone who supports our show in whatever way that you can. The Patreon page is the lifeblood of our work, so we hope you will hop on board. And that's it for me. Thanks, y'all. We'll see you next week. Bye.